Hello, I am going to tell you a story about the negative genetic interaction between the genes MCM10 and RNF4 in human colon cancer cells. So why do we care about negative genetic interactions? Well, this information can be and has been exploited to develop novel targeted cancer therapies. The efficacy of PARP inhibitors, which block single-strand break repair, and BRCA-mutated cancers, which are already deficient in double-strand break repair, highlights the clinical significance of discovering negative genetic interactions. Previously, we have found that the essential replication factor MCM10 and 6568, RNF4's ortholog in yeast, display a negative genetic interaction. And since these proteins are evolutionarily conserved, we hypothesize that MCM10 and RNF4 double mutants are synthetically sick in mammalian cells. So, to explore this, we use the CRISPR-Cas9 gene editing tool to generate our mutant cell lines, and this Western blot is just showing validation of our cell lines. So we have MCM10 heterozygous mutants. MCM10 is an essential gene, so you can only make heterozygous mutants. We have RNF4 null clones, and then we have 5 MCM10 RNF4 double mutants. And when we look at their growth, we see that the double mutants exhibit a drastic growth defect. And a couple things could contribute to this. One, the cells could have a significant alteration in their cell cycle profiles, or they could have an increase in cell death. So we decided to look at cell death by using a FACS apoptosis assay, where cells are stained with propydium iodide to stain dead cells and annexin-5 to stain apoptotic cells. You can see that the double mutants have elevated levels of early and late apoptotic cells, which contributes to their growth defect. In addition to their growth defect, the double mutants also have extremely short telomeres, shown by this telomere restriction fragment analysis, which is a modified southern blot. This signifies that they have high levels of genomic instability. We also used a FACS chromatin flow assay where we labeled the cells with a nucleoside analog, EDU, to look at replicating cells, and then determine total DNA content by DAPI staining. So we can determine each phase of the cell cycle. You can see that the double mutants have more cells in G1 and less in S phase, indicating that they have a G1 arrest. And this is very different than our yeast double mutants, which actually arrested in mitosis. So, looking in the literature, consistent with the G1 arrest, the double mutants have elevated levels of 53 BP1 nuclear bodies, which signify underreplicated DNA. So taken together, MCM10 and RNF4 protect against underreplicated DNA in human colon cancer cells. So our working model is that MCM10 deficiency causes a decrease in the rate of DNA synthesis. Without RNF4, these cells arrest in G1. When we look at the amount of EDU incorporated during our 30-minute pulse of S-phase cells, we actually see that the double mutants rescue this defect in DNA synthesis. And so we propose that inhibiting RNF4 in MCM10 mutants allows otherwise illegitimate DNA synthesis to occur, but this comes at the cost of reduced replication fidelity. And this is in line with our observations that the double mutants undergo apoptosis or cell death, experience telomere shortening, and have elevated levels of 53 BP1 nuclear bodies. And so, taken together, MCM10 and RNF4 display a negative genetic interaction in human colon cancer cells, which could be exploited to develop novel targeted cancer therapies. Thank you.